Hello everyone, welcome back to the channel. Today we are going to be talking about tissue culture, sometimes called cell culture. Now, tissue culture refers to the in vitro growth of cells or tissues derived from multicellular organisms. And this is the foundation of biomedical research and toxicology testing because these methods allow scientists to grow and study cells outside the organism under controlled laboratory conditions. So you find that a lot of toxicologists use this technique to study the effects of toxins on cells before proceeding to in vivo studies. Now, the process of the cell culture technique follows this flow chart. We start with isolating the tissues or cells of interest. Thereafter, those cells are cultured or passaged repeatedly as needed, and thereafter, those cells, either during or after culturing or passaging, can be used for experimentation and assays. It is at this point that the research is done, and thereafter, any valuable cell lines that remain can be frozen and kept as cell stock. And when needed, those frozen cells can be thawed and reused. These cultures are widely used in cytogenic, biochemical, and, as we have said before, toxicological studies. Now, a vital part of cell culture technique is the antiseptic and sterile techniques because cells must be maintained for days and even weeks. Because of this, strict antiseptic conditions are essential to prevent contamination and ensure reproducibility of results. So, sterility is the most important principle when conducting research with tissue culture. And the first way to observe antiseptic and sterile techniques is with the use of a laminar flow hood, the device you can see on the screen. It is a working space for cell and tissue culture technique. It is also specially equipped to suck in the air and prevent contamination. Another part of the antiseptic and sterile technique is knowing how to take care of glasswares and plasticwares, both the ones that are reusable and disposable. This includes flasks, petri dishes, microtiter plates, pipette tips, plastic containers, and so on. Also, the use of personal protective wears such as your lab coats, your gloves, and your protective eyewear are all compulsory during cell and tissue culture technique. All of these are used together with the laminar flow hood. This is a demonstration of how the laminar flow hood is used. All the glasswares, plasticwares, and the whole experimentation is taking place inside of the flow hood. Also, for day to day use, the cabinet must be turned on for at least 5 minutes prior to the beginning of the work, and all surfaces, both on the inside and on the outside of the hood, must be kept clean and disinfected every single day using a 10% bleach solution or 70% alcohol. So you spray alcohol or bleach on every single thing that goes into the laminar flow hood before it goes in, and you even spray it on your gloved hands and rub together before you begin any cell culture technique. Now, sterilization can also be achieved by autoclaving your glass and plastic wares at 121 degrees for 20 minutes or using dry heat at 160 degrees for 2 hours. Media solutions can also be sterilized if they are heat sensitive. So the autoclave can be used for certain equipment before use. The next important thing we are going to learn about the tissue culture technique is the preparation of the culture medium. Now, the growth and maintenance of cells depend on the composition of the culture medium because this basically acts as a source of nutrition for the cells. This is the food for the cells. And common media include Eagles MEM and DMEM. These are usually supplemented with amino acids like L-glutamine. They are supplemented with antibiotics to prevent the growth of bacteria and serum such as fetal bovine serum. The medium also provides essential nutrients, maintains osmotic balance, and helps to buffer pH between 7.2 and 7.4. Now, during cell culture, after isolation of cells, the medium is added to the cells. They are arranged in petri dishes or flasks like this and stored inside a CO2 incubator. The mammalian cells are usually stored at 37 degrees C at 5% CO2 within the CO2 incubator. So during an experiment, it is at this point where the toxicologist might add whatever chemical or toxic agent and watch what happens within the 1 or 2 days the cells spend in the CO2 incubator, after which the cells are removed and observed under a microscope. Now, another important concept in cell and tissue technique is the subculturing and passaging of cells as they approach confluency. A confluency is the degree to which a cell culture surface is covered by cells as a measure of the percentage of the total area. So 20% of confluency means that about 20% of the total area has been covered by cells. 50% is half the area of the petri dish or flax covered by cells, and 70 and so on. Now, cells should be ideally subcultured as they approach 80%. Now, we have two main types of cells. They could be a monolayer cell culture or a suspension culture. A monolayer is just one wherein the cells are attached to the surface of the flask or petri dish, and a suspension culture is when the cells are suspended. Now, when the cells are in monolayer culture, the cells have to be detached from the surface using trypsin EDTA. So this helps to detach the cells from the surface, making them suspended. Now, obviously, if the cells are already suspended, there is no need to add trypsin EDTA. So that is the only additional step when a monolayer is involved. Now, after the trypsin has been added and the cells detached, media is added into a wash flask. Thereafter, the media is placed in a sterile tube and centrifuged, after which we aspirate the supernatant to remove the supernatant, and the remaining pellets contains the cells. Thereafter, we resuspend the cells and add fresh media. So the media that we have already learnt about, we add fresh media to the resuspended pellets. These same pellets can also be divided into two new flags. In that way, you have taken the number of cells from the first flags and distributed them into two flags. Cells should ideally be subcultured at 80% confluency, which is about 2 to 3 days, ideally during the log phase. The log phase is the exponential phase in cell growth where they grow very fast before they enter the stationary phase. Now, during the process of cell culture technique, genetic drift might occur and cells may begin to lose their ability to inherit traits from one successive generation to another. So, to preserve valuable cell lines and prevent such genetic drift, cells are cryopreserved using a freezing medium containing DMSO and FBS. Freezing is done gradually to avoid ice crystal damage because ice crystals can actually damage the cells. First at minus 70 degrees C and then transferred to liquid nitrogen. So, first of all, the cells are suspended with DMSO and FSB. The DMSO is what prevents the ice crystals from damaging the cells. So, they are mixed together in a cryogenic vial. Now, these cryogenic vials are placed into a controlled rate freezing container to make sure the freezing is done at a slow rate. And this controlled rate freezing container is then stored in a minus 80 degrees C freezer. After which, the cryogenic vials are removed from the controlled rate freezing container and placed into liquid nitrogen. So the cells remain inside liquid nitrogen for as long as needed until they are required. So when required, frozen cells are thawed rapidly at 37 degrees C using a 37 degrees C water bath. You place the cryovalves into the water bath for as long as needed for the cells to melt. Thereafter, the contents are transferred into a conical tube with pre-warmed material and then centrifuged. After centrifugation, the pellets containing the cells are then resuspended with culture media into vessels, and subculturing can continue from there. Another important part of cell and tissue culture technique is the determination of cell number and viability, which is crucial for maintaining consistent culture conditions. And to observe the cells for viability or count them, a hemacytometer is used under the microscope. An inverted microscope is usually used in cell culture technique. A dye known as trypan blue dye is used to accomplish counting and checking for viability. Trypan blue dye is added to the cells. Viable cells exclude trypan blue dye, whereas the dead cells take up the dye, which will allow us to estimate the percentage viability by looking at the cells that do not take up the dye, the cells that are not looking blue under the inverted microscope. Now, the cells are counted using the design of the hemacytometer. The under the glass slide of the hemacytometer are grid lines arranged like this, and at the four corners of those grid lines are 1 by 1 millimeter squares that are 0.1 millimeter deep. So we use these four squares by the edge to count viable cells. Now it should be noted that the cells are first diluted in trypan blue using a particular dilution factor which we'll get to in a minute. So let us assume that you have diluted your cells in trypan blue and you have placed them into the hemacytometer. So you count the number of cells that are within the four boxes. So if you have 23 cells here, 27, 24, and 18, you move on to the next step. Now, the next step, we need to find the number of cells per mil of our cell suspension put into the hemacytometer. And to do that, we must get the average number of cells. That's the average number of the cells in those four boxes from the previous slide, multiplied by the dilution factor used in adding the trypan blue dye, multiplied by 10 raised 4, which is the volume of the counting chamber of the hemacytometer, and thereafter multiply our result by the volume to get the number of cells. So the average for the four boxes, the four designated boxes on the HEMA cytometer, gives us a total of 94 divided by 4, 23, multiplied by a dilution factor of 10. So let us assume that we used 10 microliter of cells and 90 microliter of trypan blue. That gives us a dilution factor of 10 because we used 10 microliter of our cells. And that gives us 230. Thereafter, we multiply that by 10 raised 4, which is the volume of the counting chamber. In other words, when you count, you're actually counting the number of cells in 10 raised 4 mil of the suspension. And thereafter, we multiply by the volume that we added into the hemacytometer to get the total number of cells. So let's just assume we put just 1 mil or 2 mil of our cell suspension into the HEMA cytometer. That would just be this 2.3 million per mil multiplied by 2 to get the actual total number of cells. And this is how you count cells. Remember, you are counting the viable cells that did not take up the trypan blue dye. Now, there are some common contaminants that affect tissue culture technique, particularly bacteria and fungi, because they cause turbidity and pH shift, while mycoplasma infection is much harder to detect and can alter cell metabolism and chromosome structure. Another thing about the mycoplasma is that this bacteria lacks a cell wall, so it is naturally resistant to a number of antibiotics. So the antibiotics in the media would not combat against it. So regular microscopic checks and mycoplasma testing help to maintain culture integrity. This is another reason you should check your cells very often to avoid the growth of bacteria and infection. And if seen, the cells must be discarded immediately and the experiment should be repeated. So let's recap on some of the things we've learned about tissue culture. We must maintain sterility by using our personal protective equipment and observing antiseptic techniques. We must use the appropriate medium to ensure that cells receive required nutrients. We must subculture regularly, preferably within 2 to 3 days, to avoid confluency and crowding in cells. We should freeze for storage using liquid nitrogen and thaw rapidly for recovery when cells are needed again. And we can monitor viability and contamination using trypan blue dye, a hemacytometer, and an inverted microscope. In summary, tissue and cell culture technique allows controlled growth of cells for research and diagnostics, and its success depends on observing all of these things that we have recapped. With this technique, we can model biological systems in vitro and advance toxicology, cancer biology, and molecular medicine. So, thanks for watching. Please give this video a thumbs up, subscribe if you haven't already, and let me know in the comment section if you have any questions or if you would like me to have or to make a special video dedicated to counting of cells for cell viability. I'll see you guys next time. Bye.